One of the most amazing things about sturgeon is that they are octoploids, meaning their ploidy is 8N or 8 chromosomes per set. As part of their work at the Nechako White Sturgeon Conservation Center, staff must test every sturgeon's ploidy. This is one step to ensure the healthiest fish have the best chance for survival. So how do you test for ploidy in a sturgeon? Well, you look at their genetic material. And where can we find their genetic material? In blood cells. The first step is to collect blood from each individual fish. To reduce harm and stress from handling, sturgeon are put in a bath of anesthetic water. Once the fish are calm, staff find the caudal vein, which is found before the tail fin on the underside of the fish. The blood sample is collected using a fine-gauged needle, strong enough to get through the tough skin of a sturgeon. Each blood sample is placed into its own vacuum-sealed tube. The tube is labeled with the sturgeon's unique pit tag number. This process takes several days to complete. The blood samples are then sent to a diagnostic lab where specialized equipment is used to analyze the DNA in the blood samples. The results are then sent back to the hatchery. The lab technicians estimate ploidy by looking at how much genetic material is in a cell based on size differences. More DNA usually means a larger cell. Think of it like guessing how many books are in a sealed box without opening it. The books represent the genetic material and the box represents the cell. A bigger box will likely have more books in it. The most common ploidy error is 12N, where there is approximately 50% more genetic material than 8N. Ploidy sampling from a conservation aquaculture perspective ensures that only 8N ploidy hatchery-raised sturgeon are released. This process protects the wild Nechako White Sturgeon population and maintains healthy genetic diversity for future generations. If you haven't already, check out the animated video about the genetics of sturgeon ploidy.